Thanks a lot for the invitation and for the very nice introduction. So before starting to dive into the microbes, I just want to spend a couple of words of where I come from. So this is where ICTP is. That's where Trieste is, in the last tip of Italy, in the Northeast. And this is a very interesting institute because it's a UNESCO Category 1 institute, which means that we have a double mission. One is to do research and the other one is to foster science in the whole world. So we have a lot of programs for scientists from all over the world. So we organize many conferences, activities, both in Trieste and literally everywhere. And so if you are interested getting involved, follow what we do, I mean, check it up, and it's in a quite nice place. So the talk today is about microbes. And I mean, one fascinating aspect about microbes is that they are really everywhere. They are able of incredible adaptation where they can survive in very extreme environments. I don't have to convince you probably that they are extremely important for many processes, from biogeochemical cycles to the functioning and the health of host they inhabit. And one thing that we know very well is that they live in very structured communities, right? So this is one example that a person with the original scene of being a physicist like a lot is Winogradsky column, where you have basically a gradient of oxygen and sulfur, and you can literally see the stratification of communities in different trophic levels, performing different reactions on the redox ladder. And sort of the same phenomenology happens when you look at these hot springs, and you have a gradient of temperature in this case, right? And so this is like a very nice example on how you can really see with your naked eye a very nice structuring of an ecosystem, right? And then, I mean, now, I guess 30 years ago, sort of the sequencing revolution came into microbiology. And so basically what people started doing is to go into these beautiful places and sampling these communities, sequence what's there, and identify which are the players in this complex community. And so what happens is that you go from these beautiful colors to these very nice places to not having enough colors to represent the diversity of these communities, right? So where really the diversity and the variability, it's what strikes everyone that start working with this data. And of course, this variability, it's something that reflects several processes, and it what makes studying this complex community particularly challenging. So if you track the community composition over time, you have to face two, I believe, fundamental challenges. One challenge is that the environment where this community change and where this population grows and decline is extremely high-dimensional, right? The number of factors that influence the growth of every single bug is extremely high-dimensional, right? So you cannot really hope to measure everything, right? And the other challenge is that the trait space, so the characteristics of the organisms that determines how they respond to these changes is also extremely high-dimensional, right? So the challenge is how do we sort of connect all these things together to understand what's actually going on in a predictive way. So the standard, let's say, one standard approach of microbiology is to try to find some hidden low dimensions in this data. What does it mean? It means that you try to focus on some environmental gradients, some axes of variation of the environment, and some corresponding traits in the population, and you can sort of connect within this mess of this complex community with all this color, a variation in some relevant traits as a response to a variation of the conditions, okay? The second approach, and is the one I will follow, is to focus more on macroscopic statistical patterns. So in some sense, instead of focusing on the whole complexity, really look at the trajectory and the structure of the composition and try to describe it quantitatively and try to understand whether there are regularities in their property, and from this regularity trying to infer properties of their dynamics, right? And so focus on what is the statistical structure of the variation. And so my talk today is really about, it's following this approach, and I think these are two equally good alternatives, so that serve sort of different purposes. And sort of the question in general that motivates the presentation is what is actually constraining the variation we observe. So if I want to generate an in silico dataset here with some colors, and I want this to be as statistically similar as possible to the data I observe, what are the rules that I should feed into my model? Okay. And so what is constraining the variation I see, and what is the meaning of this variation? What are the processes that determine this variation? What is actually signal and what is actually noise? Okay. So this is the outline of the talk. So it's divided in 3 parts. So the first part is about introducing some simple macroscopic patterns that are quite general across microbial communities from different systems and provide sort of a background or a lens on which to I believe, to study the variability of these microbial communities. And then I'll, in the second and third part, I'll describe how these patterns and this sort of simple dynamics breaks down in long human gut microbiome time series. And in the third part, how actually this also breaks down in experimental communities, revealing alternative stable states and as a fundamental mechanism, sort of making this simple approach wrong in some sense. So let's focus on the first part. And in some sense, very generally, the data that in community ecology we typically have are data like this, right? Where we have several species that can be amplicon sequence variants or OTUs from your sequencing experiments across different communities or over time, right? And the typical question that we ask when we try to model these communities is try to ask what is the probability of observing, one question that you can ask, but is how can we control the composition? So if we have a given environmental input, what is the composition of these communities? Or you can also reverse it, right? If you have the composition of this community, what is the environmental output, let's say? This is what typically the object we would like to describe if we try to model communities, right? And as I said, the question that I want to ask is what are the minimal rules to reproduce the community composition and variation? And so instead of focusing on this property here, I will focus on something a little bit simpler. So instead of asking how the composition depends on the environment, I'll ask what is the probability of observing a given community in a given state, okay, with a given composition. So what I want to build, it's a statistical model, and then using the statistical models to sort of define minimal community dynamics on how these communities are structured, okay? So an example that some of you might be very familiar with and some of you might not know at all, but if you don't know it, it's not important, that fits in this framework is neutral theory, right? So neutral theory basically assumes that all the individuals are ecologically equivalent, and what it means is that you basically assume that this composition here is just a property of what are the list of abundances and not who is particularly there. So the approach I'll describe is non-neutral, but this is just an example for the ones of you that know neutral theory to sort of frame the question I'm asking in this context. And so when you look at the data, you start to find actually remarkable path. Where you look at the data under this sort of approach, you start to find remarkable regularities. So for instance, this is like the distribution of abundances of the typical distribution of abundances of individual species across communities or over time. Okay, so you take a species and you ask what is the probability that a species will have this given abundance in a given point in time or in a given community. Okay, and you ask what is the shape of this distribution. Okay, and what you find is that this distribution has remarkable regularities is always a gamma distribution across systems that are completely different from each other, right? The top are very different systems, and you can see that they come from sort of all over types of systems. The second one is different host, different humans, and the data refers to their gut microbiome over time, okay? So you see this regularity, what does it mean? Well, one sort of the interpretation that we have of this regularity is that this reflects a very basic dynamic properties of these communities. Okay, and the two dynamics properties which are captured by this equation reflect two fundamental forces in ecology. The first one is that growth is limited. So if you have a population, this will not grow forever, but there is some limitation by resources, by other factors. And this is one part. And the other part is that the environment continuously fluctuates, right? One critical aspect is that in whatever natural communities you are observing, the environment is never static, is under constant conditions because the populations are also changing, are dynamically changing the environment themselves. And so this gives rise to constant fluctuations. And so this very simple model predicts exactly the distribution I described. So shows what you observe and also predict some simple dynamical properties of the dynamics. Basically, if you take a random species, it tells you what is the probability that a species with a given abundance today, on average, what is the abundance tomorrow, or what is the variance of the abundance tomorrow. So simple statistical properties of the dynamics of these communities. Okay? So basically, the idea is that we have this bunch of trajectories, and these trajectories are all around a given value, which is determined by this sort of effective carrying capacity, and they fluctuate because of constant environmental fluctuations. This is like the sort of zeroth order explanation of the dynamics you observe. And then you can ask for other regularities. For instance, you can ask, okay, I have different species. How do they differ in their typical abundance? Do they oscillate around the same value? Are very different in their abundances? And what you find is another remarkable regularity is that basically there are the distribution of the typical abundance follows a normal distribution across very different systems. And also you can ask what is the variability of these abundances, so the role of fluctuations. And again, what you observe is remarkable regularities on the fluctuations of this abundance, which basically tells us that all these parameters appearing in these equations also have somewhat reproducible patterns across systems. So they come from underlying processes which are pretty general across systems. Okay, and reflect somewhat general properties about resource limitation, or in general the existence of limiting factors and environmental fluctuations. Okay, so this was part one, which was pretty short, and the key points here is that there are reproducible patterns of variation. So where we look at community under this lens, we find something that is very reproducible, quantitatively reproducible in general. And basically, the interpretation is that these properties that are very reproducible in general are determined by growth limitation and environmental fluctuations. Okay? Then, of course, one could ask, is this the whole story? Should I just go out of the room and then the talk here? And the answer is, of course, not. And actually, what this provides is a framework, is a wrong model in some sense that is good enough to capture many properties, but it's something that we can use to actually interrogate the data with a solid statistical and quantitative base. And so the first question, I mean, one of the first questions that we asked, I'll try to answer using this model, is to ask how much this view of communities as collection of populations that fluctuate very much, but around a constant value is actually correct, right? If we look over long time scales, do we actually see that different populations fluctuates around a constant value for a very long time and nothing changes, right? So one simple way to ask this question is to ask how much the parameters that we inferred, this sort of typical abundance and the amplitude of the fluctuations, change over very long times. So you can take a very long time series. These are human gut microbiome data for a length of about 1 year. You divide the data in two and you ask how much the first half is correlated to the second half. You find that there is some correlations. There are also some interesting deviations, and the noise is a little bit more messy. In general, you find some correlations. And the question is, why don't you have correlation 1? Why don't you have correlation 0? Can I sort of explain, can I sort of dig a little bit in and understand what's actually going on over this time scale? And so one naive expectation that you might imagine is the one depicted on the top, that basically you have this species, this population, this fluctuation fluctuates a lot and fluctuates around a stable value, right? So there is a lot of dynamics, there is a lot going on, but it's moving along to stay in the same place, right? And so if you do that, what you would expect is that if you compare a community now and a community in the future, more or less there is not going to be an effect on how distant in the future, right? If you perhaps pass the time scale of a week, my gut microbiome today and my gut microbiome 1 month from now is as different as my gut microbiome 1 year from now, 10 years from now, okay? If this picture is correct, right? The alternative is that actually this is not stable. If you look at a time scale which is so short enough, it looks like that is stable, but is actually changing over time. And so if I look at my gut microbiome composition 1 month from now or 1 year from now, it's very different, right? Because over longer time scales, I can detect a difference. So the question is, we have a baseline model, which is like fully on the left, and we can ask, does this actually explain the trajectories we see, right? And we can sort of classify individual populations based on whether they fit with the model or they don't fit with the model. And what comes out is that we have two classes of populations. We have populations that most of the populations are the ones in red, which basically, after a short transient of a few days, they look the same over a long period of time. And then you have others that actually changes and display this non-stationary behavior. So they are very different in the future compared to what they are now. And here there are different hosts, and what you see is that most of them, most of the species, they have this stable behavior. So okay, the stable behavior, it's in some sense uninteresting. So the question is, how do the species that are unstable look like? What are their trajectory? And the way their trajectory look like is basically like this. This is a typical example where you have some species that fluctuate constantly around a value and then rapidly switches. Okay, switches and fluctuates around a given value. Okay, so this is the typical way in which these populations are unstable. And so you can track what are these times where this transition occurs. And what you see is that these times sort of cluster. So you have group of species which are fairly small. I mean, if you see the y-axis is like 10 to 15 OTUs out of hundreds that switch around some typical times. They don't switch all the time, okay? So the question is, okay, we see these dynamics. How do we capture it? So we did something, again, the same spirit of the model I described before, to sort of build a phenomenological version of the model, which basically assumes that this carrying capacity switches over some discrete times, okay? So each species has a characteristic abundance, but then can switch between alternative states. And the key aspect of this data that somewhat puzzling to me is that if you track how much these switches occur, is completely independent of the typical abundance. So very high species that are very, very high abundance, they can switch to alternative states with the same rate and with the same magnitude, relative magnitude, as species that are very rare. So it looks like the abundance of a species, it doesn't influence the rate at which they have this sort of binary switches between different populations. Okay, so then with this model, basically what we do is that we can capture what we see over longer time scales within a given host, right? So we take a given host and we can sort of reproduce the same statistics, right? Which is not entirely surprising because it's what we fed into the model. The more surprising thing is that then we can ask, now suppose that I take two different hosts, right? And I ask how different they are, right? There are two factors that contributes to this difference. So if I look at my gut microbiome is going to be different today and 1 week from now because I have these very rapid fluctuations, and it's going to be different today and 1 year from now because I have these longer term switches. Okay, so what we can say is we have a baseline expectation of how two different individuals are different, as if they were two independent processes. Basically, they are independent in the rapid fluctuations and are independent in these more long-term switches. And if we do that, we are basically able to recapitulate most of the variability that we observe across individuals, across hosts. Okay. So sorry, it's very hard for me not to have questions during the talk because in physics, this is so I don't know if I completely lost you. I'm trying to guess the faces and slow down. Yeah, but okay. So, but the summary, I mean, what you should take of this second part is that the first part is almost correct, but is in a good part wrong. And is wrong because there are, if you look at the long time scales, there are very abrupt, rapid shifts of abundances. Of the typical abundance this population fluctuates about. And interestingly, these switches are not independent across species, and we don't fully understand what is control that. And they recapitulate most of the variability between hosts. So actually, most of the variability between hosts is because the dynamics of our own microbiome has this short time scale of rapid fluctuations, but also this very long time scale on the scales of months, of years, of these switches, alternative stable states of the abundance. So the question that came out of this is, I mean, there are many questions, is what is the source of these alternative stable state or switches? One natural question is whether they are sort of endogenous or driven by some change in the habits of the host. Perhaps there is a typical time scale in which we change, I don't know, diet or activity or stress level or whatever. Are they fully endogenous or are they driven by external factors? And the other question which you might have is how this changes if we look at different levels of organization, right? Right now, I've been talking about species between quotes, sort of talking about amplicon sequence variants or OTUs, which are very operational way to define species, right, for microbes, but they might not reflect at all the relevant scale of ecological role and ecological function. So what we decided to do is to focus on experiments under very controlled conditions and study alternative stable states in those contexts. So the experiments we consider are experiments performed by other groups, which basically did something in some sense very interesting and boring, which is to take a diverse pool of species from some natural sample, which can be soil or leaves, and then inoculate some defined medium with this sample and transfer it for a good amount of days, every 48 hours, and then look at what is the community at the end, or actually the community during these dynamics you can do. And then you can do it in replicated condition many times. And so look at the variability of these trajectories of system that has been assembled by the same inoculum, is growing in the same condition over a trajectory. And the question is, in this very controlled condition, what is the variability? How does it look like? Okay. Sort of the common wisdom in this in the structure of the variability in this kind of setting, which is also like sort of typically considered in natural system, is that somehow they exist two different scales or two different kind of sources of variability. There is, if you look at a very fine, if you look at a fine level of fine taxonomic level, for instance, you look at species composition, you see high variability. But if you look at a coarser level, and typically it's considered family level in these kind of experiments, you find something that is very reproducible. So this means that if you take this experiment and you grow these bacteria on, let's say, glucose as main carbon source, you find a lot of variability in the composition at the species level, but at the family level is very reproducible. So if you now grow them on citrate, you change the family composition in a consistent way, while the species level varies a lot. Okay, so this is one aspect. And the other aspect is that actually the picture at the family level is a bit more complicated, because not only you have this variability, you also have alternative stable states. So actually, if you look at the family level with these highly replicated communities, you see different family groups that sort of corresponds to different traits, let's say, related to being fast grower, fast respirators or fermentators. Let's say you have different stable states of the assembly of the community. You have different composition, even when assembled under the same condition, which is reflected at the family level. Okay? And so the question that we asked is, okay, this is like sort of telling us that what we see at the family level is where the interesting ecology happens, and whatever we see at the more fine-grained composition, it's somewhat driven by noise, contingency, very hard to reproduce. And so the question that we have is, this common wisdom is that there are, let's say, alternative stable states at the family level, which actually reflects the interesting ecology. And then we have somewhat a notion of functional redundancy, where there are multiple species within the same family that are sort of ecologically overlapping in their function. And there is like small or minute source of variation determine whether there is more of species A or species B within that family. Okay? And so this, in some sense, should give strong explanation of the variation we see at the fine level. So what we did was, let's look at what are the patterns of variations inspired by what we did in natural communities in this community here, where we have a lot of data. And so what we found is something quite different from natural communities in this setting. And basically, one clear observation is that when we look at individual species, in this case, individual amplicon sequence variants, if you know what they are, is that consistently their distribution, their abundance distribution is bimodal. So they have two different peaks, right, consistently. So the vast majority of DSVs across these different experiments, they are bimodal. Okay? And then we said, okay, perhaps these two modes are sort of independent realizations of the patterns that we described earlier. And what we see is that basically the patterns that we saw earlier hold independently for the two different peaks. So basically, it's like, and the two value of abundance of the species are completely independent, can be whatever. So meaning that a very high abundant species, the low abundant peak is way more abundant than the high abundance peak of a low abundant species on average. This is the part that for me it's very hard to say, but I said it. Okay, meaning that this is a low abundant species and the high peak is sort of at the same value or lower than the rare peak for the top species. So these two things are like sort of typical of each species, and they are not like sort of ordered in an obvious way. And the sort of patterns of variation that we saw earlier. So what is the variability within each peak is similar to what we observed earlier. So it holds also in this case. So basically, the sort of picture that emerged from this data is that it looks like there are actually two sources of variation, right? Capturing at this very fine level. So there is one source of variation, which is this within state. So a population can be in these two states, high abundance or low abundance. And the variation, and then there is variation within. Now, one question that you might have, that I'll answer now, is, well, perhaps the low abundant thing is basically something at the border of being absent, or is something that is actually declining and going extinct. Actually, we have time trajectory, so we can see that there are sort of dynamic switches between these two states. So it's really not that these are just things that are absent. Okay, so it's really there are these two states. Okay, so basically we have these two sources, and then the second question that we asked is, okay, so if we look at this variation, this will be determined by interactions between the members of these communities, since the external conditions are fixed, right? So it has to be the interaction between the members of this community that somehow determines whether this by stability. So what we try to ask is what in this picture of variation reflects interactions. Now, of course, we don't have a way to measure or infer interactions directly. The only thing we have to access are the correlation between species, right? So sometimes we can ask whether a species is high, is the other species high in the same community? And then you can do something very simple. You can look at the distribution across pairs of these correlations, and there are correlations which are don't occur by chance. So these species are not fluctuating independently as expected. And so if you shuffle the data, you get something very different. The point is that when you discover these two sort of contributors, statistical contributors of the variation, both within variation and between variation, both contribute to the correlations, right? So whether species is in the high or low abundance, together with another species contributes to variation, but also when both are in the high abundance state, whether their abundance is correlated, contributes to the correlation. So you have these two different contributors, right? And so do they both contribute? Is there a signal in both about the interactions? And what we discover is that actually the correlations that we see in the data are completely reproduced if we preserve the abundance state and we shuffle the abundance within. What does it mean? Is that basically it looks like we have two types of variation, this between state variation and within state variation. And the within state variation is weakly correlated across population, and sort of seems to reflect somewhat very contingent processes occurring on very rapid time scales. And there is something that is setting the typical value of abundance. So this is like sort of consistent to the picture of the first part, basically. And then the between state variation is actually where there is interesting ecology, which is very structured and sort of corresponds to different degrees of limitations. So the question is now, okay, we see that there is where we should focus on. What is actually the structure there? What does it reflect? So then basically we can forget about the actual abundance, and we can just focus on whether a species is in the high abundance state or the low abundance state. That is where the key information lies, right? And so then what we can ask is, if we take two communities, right, are they similar or different based on the state where each species is. Okay? And when you do that, and you compare communities, you find that you have a remarkable block structure. What does it mean? That you have communities that are very similar to each other and different in similar ways to other communities. This was the second hardest thing to say. And we sort of identify 4 blocks, right? So 4 typical communities which are very similar within and different in similar ways to other communities. Okay? Some are very big, some are very small, but this is like very similar to very consistent depending on the method you use to define. Okay, so basically we have these blocks, these groups of communities that I'll call attractors, and they reflect one characteristic state of the community. Yes, one characteristic state of the community. Then the question that we can ask is, okay, we have in mind that somehow so we have a way of building somehow where the information about the interesting ecological processes is based on single species variation. How does this map into this idea that actually the interesting ecology occurs at this coarse level, like family level? Okay, so one natural expectation that you might have is basically this is constrained by family level, right? So if we look at these at community within each of these blocks, then we should expect that the family is very constrained. Now, if the family is very constrained, what you should expect is that basically this is sort of fixed, and the variation within this family is constrained on the family level, which means what? What you would expect is that the variability of abundances of species within a family should be negatively correlated because you are constrained by the family, and there is no signal of that. Okay? I don't know if you are allowed. Sorry. I wish, but you can have a no. Yeah, I'll take the question at the end. Sorry. So the point here is what you expect is that if you are constrained by the family abundance, so the family abundance is actually fixed, and then you have variability within, then you would expect that species within the same family in each of these attractors are negatively correlated because they are fixed sum. And this is not what you observe. Okay, so then we say, okay, let's look at what is characteristic about these attractors here. What is characteristic about these attractors is that you can look for each attractor, how likely is a species to be in the high abundance state and this in the low abundance state. So this is basically a barcode of the community composition in each of the state, right? You look at one state and you say, okay, which species are going to be in their own high abundance state or in their own low abundance state, okay? And what you find is this, okay, whatever. But then when you look at it a little bit more closer, you realize that these has characteristic blocks. So there are groups of species that have the same prevalence in the same attractors. You don't have all the combinations, you will have many more combinations, few more combinations, but you don't see all of them. And you see that only some of them are populated, okay? So basically, and the other thing you see is that you have these groups that are sort of reciprocal to each other, right? You have some species which have some preference and some species which have the opposite preference, okay? So basically, the question that we ask is, good, so how does this map actually into taxonomy or phylogeny, right? So if we take 2 species that are within the same family, do we typically see them having the same preference, the opposite preference, right? So basically, this is like taking a compression of the data into where there is the interesting information about the variation of these communities. And the question is then, is there a taxonomic signal there? Right? And why this is interesting is because, well, you might expect that the variation between these different attractors might be driven by whether by sort of effective environmental filtering, where similar species tend to like the same environment or tend to dislike the same environment, versus competition, right? They don't want to stay in the same environment. And what we find is that actually the significant signal is in the reciprocal group. So actually species within the same family tends to have different prevalences, right? So somehow the preference for a given attractor is not something that is characteristic of a family, but is characteristic of a family whether the sort of which attractors have the same response, but is not characteristics the sign of the response. You might be growing well in one environment or growing well in another environment, suggesting that sort of the driver of these preference is actually something more akin to limiting similarity problem. I conclude. So third part, basically when we look at the variation and these the variation in these assembled experimental communities, we see that there is the interesting part of this variation is the existence of this high and low abundance state, like these binary switches, and the co-occurrence of high and low abundance is highly correlated because of underlying attractors. And there are groups with consistent attractor preferences where there is clear taxonomic or phylogenetic, in this case it's the same, signal. And closely related species are typically occur in reciprocal groups. So they typically have switching preferences. So just to take everything together, so I started with this mess of colors and having a palette which is not enough to represent the diversity of microbes. The point is that in this variation, there are statistical patterns which are very reproducible. And this variation is not noise. And it looks like that a general observation is that this variation is actually partitioned into some very rapid continuous fluctuations, which are probably rapid fluctuations of the environment, and some likely endogenous abrupt rare switches between different states. And with that, I'd like to conclude and thank all the people in my group and you for your attention. Thank you very much.